All right, Bioheads. This is our first AP exam review video for the 2019 AP Bio exam. We're going to do a little bit of biochemistry, some cell energetics, and some cell biology. Okay, before we get started, a couple of things to help you. The review schedule with videos is posted at that bit.ly link. I highly recommend you check out the College Board Learning Objectives, which you can also get to via that link. And there are Google Forms that you can use to submit questions for future videos. All right, let's get started. So 3 big categories of topics in this video that we got some questions on. First, just a couple of things about biochemistry and water. The big idea here is that life is made out of just a few elements. And so the arrangement and the shape of those things is going to be really important. Next topic, cell cycle, mitosis, and just a little bit of meiosis. We're going to do most of meiosis in the genetics video next week. And our third topic is what I call energetics, so photosynthesis and cellular respiration. You can also think about that in ecology and systems when we do the carbon cycle, but we're going to focus specifically on the processes inside cells. All right, starting out with biochemistry and water. If you look at monomers of the big molecules that make life, they look really, really similar, right? So it's carbon with some hydroxyl groups, some OHs on the sides. You see some oxygens, you see some nitrogens. These are really, really similar. Now, you are not expected to memorize these shapes or even these names for the AP Bio exam. You'll be given relevant information and asked to interpret it. So if I give you those, right, now we're looking at amino acids from left to right, amino acids, fatty acids, sugars, and then a nucleotide. And those things come together to form our big classes of macromolecules. And they have radically different properties and radically different functions inside cells. So how is that possible if they're all made of the same basic elements, right? So all of them have carbon, hydrogen, and oxygen. And proteins and nucleic acids have nitrogen, nucleic acids have phosphorus, and then some of your amino acids are going to have sulfur. I'm going to put that in parentheses. But the idea is all of this stuff is made of the same basic building blocks. So it really does come down to shape and polarity. Okay? So quick review on polarity. Water is the molecule that life exists in, and because oxygen and hydrogen don't pull equally hard on electrons, oxygen pulls much harder on those electrons than hydrogen, and so as a result, the electrons that are shared between the oxygen and the hydrogen atoms in water tend to spend more of their time around the oxygen and less of their time around the hydrogen. And so that makes water what we call a polar molecule. It's got a negative pole on the oxygen and it's got a positive pole on the hydrogen. Okay, if my highlighter works, hey, there it is. So here's our negative pole. The electrons spend a lot of their time over here. They spend a lot less of their time on the hydrogen side. Okay, so if you take not just one oxygen molecule, or excuse me, not just one water molecule, but several of them, you start to get these attractions between the partially positive hydrogen side of one water and the partially positive negative oxygen side of the next water. And so these hydrogen bonds, okay, these attractions between the hydrogen of one water and the negative side of the next water over, these hydrogen bonds make water molecules attracted to each other. So if you have a whole bunch of water molecules, you start to see this lattice, okay? This is why ice crystals, why frozen water is less dense, takes up more space than liquid water, okay? Those hydrogen bonds have to line up to make all of this work. And in fact, all of the cool properties that are true about water that make life possible. So adhesion and cohesion. Water sticks to itself. Water sticks to other stuff, respectively. Surface tension. Okay? The reason that insects can walk on the surface of water is because the surface of that liquid, those molecules are attracted to each other. Okay? That surface tension is really just a property that comes from adhesion and cohesion. Okay? Capillary action is adhesion and cohesion through really thin tubes. This is how water can travel up the trunks of trees. The reason you don't want to touch hot food right out of the oven is because your food is mostly water. Water stores a lot of heat. It's really hard to change the temperature of water. And then we know ice floats. Ice floats because it's less dense than liquid water. It's less dense because those hydrogen bonds have to form that lattice structure, that crystal structure. So you can think of these as 6 different properties, or you can think of these as 6 results of hydrogen bonds between water. And so hydrogen bonding is really, really important inside our cells. So if you remember the induced fit model for enzyme and substrate binding, right? So we have a substrate which looks like it sort of would fit into this enzyme's active site, but not quite, and then somehow they come together. So one of the popular ideas about how this is possible is that if you have a whole bunch of hydrogen bonding between that enzyme's active site and the substrate, that's going to get both of these molecules to subtly change shape, which, if you think about it, is starting to put some tension on that substrate molecule, okay, which is part of what lowers activation energy. Hydrogen bonding is really, really important in your DNA. The things that hold your nucleotides together, A to T and C to G, are hydrogen bonds. So hydrogen bonding comes up over and over and over again in getting the molecules that need to react or need to interact inside cells to do that in just the right way. All right, so student question was about cell cycle and mitosis, and to sort of compare and contrast mitosis and meiosis. All right, so we're not going to get into the details of sort of the genetic variation implications of meiosis. We're just going to focus on sorting chromosomes and cellular processes here. Okay, let's start with the cell cycle. Okay, so what you may have heard of as interphase, or what we used to think of as interphase, we're now going to talk about as 3 separate phases. Okay, so G1, S, G2 collectively make up interphase. So G1 is when your cell is going to do most of whatever it's supposed to be doing for your body, if we're talking about human cells or cells inside a multicellular organism. So G1 is when your liver cells are doing what liver cells do, right? Filtering blood, talking to the pancreas, right? Controlling blood sugar. So G1 is where the cell does what it does for your body. Okay, I don't like this notion of interphase being in between mitosis, as if that is the star of the cell cycle. You don't want your cells dividing all the time. You want your cells doing what they're supposed to be doing. So they spend a lot of their time in G1. S is when you get your DNA replication. So you can see here on the right side, we have in G1, if your chromosomes were to condense, you'd see them in that single unduplicated form. And then after S phase in G2, now you see these duplicated chromosomes. They have the same genes on them they did over here, but now they've replicated themselves. They have duplicate copies of everything. Now they're ready to divide. So remember, your cell cycle is controlled by proteins present in the cytoplasm. Okay, so we have to think back to transcription and translation, that genes are active throughout the cell cycle, being transcribed to RNA, translated into proteins, and these cyclin proteins regulate the cell cycle by interacting with enzymes called cyclin-dependent kinases. So the sequence and timing of all these events in the cell cycle is constantly being controlled by the interaction of genes and proteins. Okay, so this is a great time to pause this if you haven't looked at this in a long time. And you want to ask yourself, if I were given this diagram of these cyclins on the AP exam, what does it look like each of these cyclins do in terms of timing the cell cycle or pushing the cell cycle into the next phase? And what might happen to the cell cycle if I took one of these cyclins away or expressed it too early or too late? Which of these things seem like they might hold up the cell cycle if they were missing? These are things I'll let you think about. All right, so just want to check in a couple of the learning objectives, and this is why I say you want to go back to the College Board learning objectives because these are the test questions. So you will not be expected to memorize any of the cyclins, right? But learning objective 3.7, you can make predictions about natural phenomena occurring during the cell cycle. So if you had this graph in front of you and you saw an increase in cyclin E, what would you predict the cell would do, right? So it looks to me like this increase in cyclin E is the thing that pushes the cell out of G1 and causes it to begin S phase. Okay. You also want to be able to describe the events in the cell cycle. So G1, cell doing its job. S phase, DNA replication occurs. G2, cell preparing to divide. Okay. Just to be a little bit persnickety, mitosis is not cell division. Mitosis is not splitting cells. That's why cytokinesis is often listed as its own event. Mitosis is division of the nucleus. Okay? So it's not division of cells. That's a different thing. That's cytokinesis. It's not DNA replication. That happened back in S phase. Mitosis is nuclear division. You're taking the nucleus and copying its contents. Excuse me. Even I'm doing it. You're taking the nucleus and dividing its contents and making two of them. Okay. So, mitosis and phases. So if you ever made flashcards that have PMET on them, prophase, metaphase, anaphase, telophase, and then maybe you read your Campbell book and now there's this thing called prometaphase. For AP Bio, we don't care about the names of the phases. We care about what happens in mitosis. So, if you can start with duplicated chromosomes, you can line those chromosomes up single file in the middle of the cell, and then you can split those chromosomes apart so you end up with single chromosomes in each cell, but the same number that you started with, you're all set. So if I go back here, notice we start with 4 duplicated chromosomes. We have 2 big chromosomes and 2 small chromosomes, but we have 4 total chromosomes. Then you line them up single file on the metaphase plate, and then as the spindle fibers pull, these sister chromatids are ripped apart from each other, and you end up with 4 single chromosomes in each of the daughter cells. Okay? The key thing to remember with mitosis is the way these chromosomes are aligned down the center of the cell in metaphase, and that's the single file line. That's what ensures your chromatids are separated from each other, and you end up with equal numbers of chromosomes in each daughter cell. Mitosis is the reliable cell division that gives you exactly what you started with. Okay, So this is going to be true for mitosis and meiosis. Starting materials and products are the things you want to focus on. So here in this diagram, I've cut out all the middle, and we've just got what we're beginning with in prophase and what we're ending with at the end of the mitotic division. So you have a diploid cell, 2 copies of the tall chromosome, 2 copies of the short chromosome, and then in each daughter cell you've got 2 copies of the tall chromosome, 2 copies of the short chromosome. Okay, trying to stay out of the genetics as much as possible and just focusing on what's going on inside the cells. We'll get to genetics in a later video. All right, let's compare and contrast mitosis versus meiosis. This is a really common student question. So we're going to start with the same diploid cell in G1. Notice the chromosomes are singles, okay? They are not yet duplicated chromosomes. They're still shaped like I's rather than X's. Okay? So now, in meiosis, right at the beginning of meiosis, rather than the chromosomes starting to line up single file getting ready for metaphase, they line up with their homologs, with their matched pair from the other parent. Remember, you got half your chromosomes from mom, half from dad, and so you have this matched set. Okay? So chromosomes line up in their homologous pairs. Okay? We call them tetrads because these duplicated chromosomes, each chromosome has 2 sister chromatids. So if you have a homologous pair of duplicated chromosomes, you've got a total of 4 chromatids that could potentially cross over and exchange DNA. Okay. So you've had your tetrads form. You've potentially had your crossover. You've also had independent assortment, meaning when we get to metaphase 1, you could have this chromosome from mom on the left and dad on the right, or vice versa. Because they're lined up double file, you could have either parent's chromosome on the left and right for each of your, in your case, 23 pairs. Okay, so here's the big difference, and here's what you want to focus on. In meiosis 1, the chromosomes don't line up single file like they did back in mitosis. They line up double file. So now when the spindles come, what's going to get separated are not ripping sister chromatids apart from each other, but pulling these chromosomes to this cell and these chromosomes to this cell. And so now we're going to go from a cell that had 4 chromosomes to 2 daughter cells that now have 2 chromosomes each. Okay? So we call meiosis a reduction division because you need to end up with cells that have half the normal number of chromosomes. So we're halfway done with meiosis. We've reduced our chromosome number from 4 down to 2, but we still have duplicated chromosomes. So now we have to do meiosis 2, which looks, I'm skipping right by Mendel's law of segregation. We'll get back to that. But meiosis 2 looks almost exactly like mitosis, which is to say, take your duplicated chromosomes, and now they line up at the center of the cell in metaphase 2, and when they're pulled apart, you pull the sister chromatids apart, and you end up with daughter cells, 4 daughter cells, that each have single unreplicated chromosomes, but now each one of those has 2 unreplicated chromosomes, where we started up here with 4. So now we've reduced our chromosome number from 4 to 2, or from 2n down to n. You now have your haploid gametes, your egg or sperm cells. Okay. So remember, the big idea in meiosis, you have 2 rounds of division with no DNA replication in between. You start out with a 2n or diploid cell. The chromosomes line up single file in meiosis 1, reducing the chromosome number, and then they line up single file just like regular mitosis, and then we end up with the 4 daughter cells. Okay, I'm just going to leave this up for a minute, looking at the difference between the products of mitosis which are diploid, 4 chromosomes per cell, and the products of meiosis, which are haploid, 2 chromosomes per cell. Okay. All right. I think of mitosis as the essential but somewhat boring cell division, right? I don't say that pejoratively. The whole idea is you want it to be boring. You want it to make exactly what you started with, particularly if you're a multicellular organism. You want to make more of exactly what you have if you need to do growth or repair. Okay? So, my summary of mitosis is take your single chromosomes, duplicate them in S phase, get ready for mitosis in G2, and then split them up. Okay? In meiosis, you start out the same way, duplicate your single chromosomes in S phase, then separate the pairs in meiosis 1 and separate the sister chromatids in meiosis 2. I tell my students everything interesting about meiosis, everything that produces genetic diversity, happens in meiosis 1. That's where you get your crossing over, that's where you get your independent assortment. Okay? All right. Okay, last topic for this video, photosynthesis and cellular respiration. What do we really need to know? And then a couple specifics about cellular respiration, anaerobic respiration, or fermentation. Okay, I really, really like these two overview images. These are both from the Campbell textbook that so many AP Bio students use. I think these overviews show us some similarities between photosynthesis and cellular respiration, and they give you most of the detail you need to be thinking about for AP Bio. Okay, so first, and I'll come back to this sort of similarity between photosynthesis and cellular respiration in a little bit, but the first thing to notice is that these two things, these two processes, both take place in sort of bean-shaped organelles in the cytoplasm of eukaryotic cells. Okay? Chloroplasts in plant cells, mitochondria in just about all cells. We'll leave bacteria out of this for now. Mitochondria in all eukaryotic cells. So it's got a nucleus, it's got mitochondria. That's where you're going to do most of the processes we associate with cellular respiration. Okay. Similar structures, similar processes. That's an interesting thing to take note of. These chemical reactions are pretty similar to each other, and the structures look very, very similar. Okay, photosynthesis. I really like this a lot. When you see photosynthesis, draw yourself a line between photo and synthesis. The photo part are the light reactions. Okay? So the light reactions have nothing to do with carbon, nothing to do with sugar. If you look at this diagram, what's going into the light reactions, you have water, which is being brought into the plant cell. If we're talking about trees or big terrestrial plants, right, we're bringing water up through the stems to get to the leaves. So water comes into the chloroplast, and then in these stacks of thylakoids, you have the light reactions where the energy from photons of sunlight is used to split this water up. And then the electrons that were holding this water together, remember those electrons that are unequally shared makes it polar. Those electrons are then, those electrons are used to charge these two molecular batteries. Okay, so you have NADP plus and ADP, adenosine diphosphate, and if you put extra electrons onto those things, you end up with NADPH, and you can add a third phosphate to ADP, making it ATP. So it is not an oversimplification to call these things, molecular batteries. Okay? A battery holds electrons. That's what these things do. And if you have a charged battery, the ATP or the NADPH, you can use that stored electrical charge to do stuff. In this case, the stuff we're going to do is the Calvin cycle. So we're going to take carbon dioxide from the air, and through a series of chemical reactions, we're going to smash those carbons together, and eventually we're going to get some sugars from it. Okay? So photosynthesis, we have the light harnessing part, and then we have the synthesis part. The synthesis part is where we're actually going to build our sugar. And the synthesis part is electrically powered. The purpose of the light reactions is to charge those batteries. Okay? All right. Now, I said a minute ago, the electrons are coming from water. Well, when we look at some of the detail of the light reactions, that's not where we think of them as being stored, but ultimately that's where we're getting our electrons. Okay? So we'll come back to the details in just a minute. But before we get to details, I want you to keep in mind this overview. The light reactions charge these electron carriers, and then the Calvin cycle uses those electron carriers to power this manufacture of sugar. Okay? If you can stick with that idea, that's most of the detail you're going to need for AP Bio. Okay, cellular respiration. Sort of like photosynthesis in reverse, right? Now we're starting with sugar and we're ending up with CO2. But if we look at this, the steps look a lot different. So first important difference between cellular respiration and photosynthesis, the first part of cellular respiration does not happen inside mitochondria. It happens in the cytoplasm. Okay? So in the cytoplasm, you take a 6-carbon glucose sugar, and it's broken down into 2 3-carbon pyruvates. Okay? Doing that, you end up making a little bit of ATP, and you charge one of those electron carriers, NADH. So that's good, I guess. You've made a little bit of ATP. You've charged this NAD to NADH, so now you have some electrons that you can do some work with. The only downside is you've now made this incredibly poisonous compound of pyruvic acid in your cytoplasm, and you're making this all the time. So you need to do something to get rid of that poison that's floating around in your cytoplasm. So if you're a eukaryote, and we are, which is awesome, you get to take that pyruvic acid and it's transported into the mitochondria, and then the second part of cellular respiration is something called the Krebs cycle. And without looking at the details of that, it seems like the Krebs cycle makes another NADH, so it charges another battery, and it makes a little bit of ATP. Both of those things seem pretty good. Now, the Krebs cycle is where the remainder of your carbons are spun off and released as CO2. Okay? So interestingly, we think of CO2 as being one of the byproducts of cellular respiration. Not a byproduct of glycolysis, and it's not a byproduct of that third electron transport chain step. It is a byproduct of that Krebs cycle, okay, which happens in the matrix of the mitochondria, or if you think about it, the mitochondria has 2 membranes. It's got an outer membrane, and then it's got this squiggly inner membrane. The Krebs cycle happens all the way inside the squiggly inner membrane in the matrix. Okay, for just a second, let's look at cellular respiration in way too much detail. I think cellular respiration and photosynthesis are a place where we in AP Bio can get a little bit detail obsessed. So I'm going to try and break you of that in one slide. Okay? Glycolysis is not a chemical reaction. It's a series of 10 chemical reactions that get you from your starting product of your starting material of glucose to your product pyruvate. So it's really 10 chemical reactions. And then you take your pyruvate, that's transported into the mitochondria, and then the Krebs cycle, also called the citric acid cycle, we're all sorry, is a whole series of chemical reactions that reorganize carbon molecules. And as you do that reorganization, occasionally you reduce some NAD to NADH. So there's one, there's another one, which is nice. So we are charging those batteries. We're making some ATP, which is great. This is more detail than you're ever going to need. All right. And when we look at this much detail, we sort of lose the big picture. Okay. So let's simplify this a little bit again. Now we're back to something I recognize. I've got a cell. My cell has some cytoplasm. My cell has mitochondrion. Okay. Glucose comes into the cell. Glycolysis happens. Now instead of being 10 chemical reactions, it's this nice pink rectangle. It makes some ATP and some pyruvate. If you have oxygen, if you're doing aerobic respiration, you can take that pyruvate and dump that into the mitochondria to do the Krebs cycle or the citric acid cycle, whichever you want to call it. The citric acid cycle charges some more NADHs. There's another electron carrier called FADH2. And then those things go to the electron transport chain. So now you're taking those electrons off of those batteries and you're using them to do a bunch of work. And I'm going to show you that in detail in just a second. But we had a student question. What is the deal with anaerobic respiration? What is the deal if I don't have this oxygen and thus I cannot do the mitochondria related steps? Now, instead, you have to do fermentation. Okay? So, we animals do lactic acid fermentation. Okay? So, one version of fermentation, we make lactic acid out of our pyruvate. Okay? It's still not good for you, but it is significantly better for you than pyruvate. Okay? But you could do, let's say if you are a fungus, okay, you could do, let's say yeast, for example, take your pyruvate, and instead of doing lactic acid fermentation the way we animals do it, there is this other pathway that makes, instead of lactic acid, it makes ethanol. So ethanol fermentation is how we get alcoholic beverages, beer, wine, etc. Right? We take sugars in liquid and we feed them to a bunch of yeast. The yeast eat them. They do this anaerobic respiration. They do this fermentation, and they produce this alcohol in the grape juice or the wheat juice or what have you. Okay, so at long last, I want to look at the electron transport chain in mitochondria, which is here, and then the electron transport chain in chloroplasts, which is here. Okay, so if we're dealing with mitochondria and cellular respiration, here's where our Krebs cycle was happening. That's really messy. I'm getting rid of it. Okay, your Krebs cycle is happening all the way inside in the matrix, and you're making a bunch of FADH2 and NADH. And then you take the electrons off of those charged batteries, and then those electrons are passed from one protein to the next, to the next, to the next in an, wait for it, electron transport chain. So you have a movement in this diagram from left to right. You have a movement of electric charge. Movement of charge, if this were a physics class, we would call that current. You have electricity. This is a circuit. Okay? And so this electrical current moving is a form of energy, and that energy can be used to do work. In this case, as those electrons are passed from one protein to the next, an H+, also known as a proton, that H plus ion, that proton, is pumped through the membrane, so that's facilitated diffusion, okay? Not through a cell membrane, but through this squiggly cristae membrane inside the mitochondria. That proton is pumped from where we have the Krebs cycle in the matrix out into the intermembrane space, and you end up with this concentration of protons. Okay, so that's what the electron transport chain does. It uses the electrons that we stored on NADH and FADH2, and then it takes that energy and it uses it to pump a whole bunch of protons out into the intermembrane space. Okay, so this only works if you have oxygen, and here's why. At some point, if these protons are pumped out here and we've used these electrons, and now these electrons end up on this last protein in the chain, at some point we're going to need something to complete the circuit, to steal these electrons off of that last protein. That something is oxygen. And so, if you're breathing in molecular oxygen, the O2 gas, that oxygen will steal those electrons, and then it will bond to some of these free protons to make water. So this is why if you ever exhale onto a cold window, you get all that condensation. You're exhaling a bunch of not just CO2, but also H2O. All right, final step. We've now set up this concentration of protons in the intermembrane space. They fall through this protein channel, my favorite transmembrane protein of all time, ATP synthase. And so this thing works just like a water wheel. As the protons fall through it, it starts to spin, and as it spins, that mechanical energy takes that third phosphate and bonds it to ADP, so now you have your ATP. And then the majority of ATP made in aerobic respiration is made in this last step. This is the big payoff. This is why we did all of that charging of batteries in the Krebs cycle. Okay, so you may be wondering at this point, why did I put these two images together if I was only going to talk about cellular respiration? Well, the reason, if we look at this membrane with some proteins stuck through it and an ATP synthase, and then we go over to look at the chloroplast, we have this membrane. Okay, this is the thylakoid membrane. We have a bunch of proteins stuck through it, and we have an ATP synthase. These two things look remarkably similar. The only real difference here is that the chloroplast starts with a photon of light hitting a chlorophyll pigment. Okay? So remember, the center of your chlorophyll pigments here and here. The center of those is a piece of magnesium metal. And so, being a metal, if you hit it with enough energy, it will excite an electron. And that electron will fly off and then be accepted by these electron-hungry proteins, just like we had in the electron transport chain in mitochondria. And so, as these electrons are passed from one protein to the next, we end up charging NADP to NADPH. And in this case, in plants, we need water to donate some new electrons to recharge that magnesium metal. And what's left over after you've done that, if you take the electrons out of this water, you end up with some oxygen and some hydrogen. The hydrogens are going to flow through the ATP synthase, just like they did in cellular respiration, and the oxygens are the byproduct. Okay? So the reason we say oxygen is a waste product of photosynthesis is because it was only getting used so that the water molecule could donate its electrons. So, looking at these two things, remember, these are proteins stuck through membranes of these organelles in living things. And this incredibly complex electrical circuit made out of proteins. You've got to ask yourself, what are the chances that evolution built this thing twice? Doesn't it seem much more likely that these incredibly intricate electrical circuits came from some single common ancestor? So the incredible intricacy and similarity of the electron transport chains in chloroplasts and mitochondria is powerful evidence for this endosymbiosis theory, this idea that chloroplasts and mitochondria came from some original ancestor where one bacterium engulfed another one, and that interior bacterium became a specialized organelle. Okay, so, and off the top of my head, I can't tell you whether the chloroplast or the mitochondrion came first, but this idea that they must have some common ancestral process. I look at these two circuits and I think these things had to come from one origin. Okay, I think that's probably enough for one video. I want to just remind you, if you have other questions on other topics, please check out the review schedule. It's posted at that bit.ly link there. Any questions on this video or anything that I didn't think to put on the review schedule, please shoot me an email, steven@mrtrapagan.com. Thanks. I hope this was helpful. Bye.